Hello, everybody. My name is Jean-Luc Freyken. I'm co-founder of Spectradyne, and I want to thank you all for joining our webinar today on April Fool's Day. Hope everybody is doing okay with the COVID virus pandemic. These are wild times for sure. We are hanging in there, working from home as best we can, and I imagine many of you are too. So thanks for joining us. All right, so thanks again for your time. The webinar today will talk about accurate particle analysis and why that's important for good control of your science and making sure that you can tighten your experimental variability as much as possible. So in the webinar, we'll talk about three basic sections. One will be why particle concentration is so important. And then two, we'll talk a little bit about Spectradyne's technology, microfluidic resistive pulse sensing, and how it delivers more accurate particle concentration than other techniques. And then we'll hear from Zach Troyer from the Tilton Lab at Case Western Reserve University. And he'll describe a case study where using, being able to obtain accurate concentration enabled important improvements in their extracellular vesicle isolation methods. Then we'll open up to answering questions from any of you. So to ask questions, there are a couple different ways. You can either just type a chat message into the Zoom feature, look for this icon, or you can shoot Lou an email directly at this email address. I'll give you a sec to type that down. Okay, so why is concentration so important? And the punchline essentially is that particle concentration is a critical experimental variable, and we'll go through a couple of examples of highlighting that. And so, controlling your experimental variables leads to tighter outcomes, right? Which is what we're all after as scientists. So one common case is people using particles as therapeutics. And so here we're looking at an example of, for example, using them for drug delivery. Maybe these are EVs, maybe these are liposomes, or other therapeutic containing particles. And in this case, concentration is critical because it's the dose that's being administered, right? So either you're trying to control the safety of your product, or maybe you're comparing two different formulations and you're testing these in a test system like a mouse or cell culture. Obviously, no one would consider comparing two formulations without first controlling for the amount of drug that's being administered to both test systems. So the concentration, measuring the concentration properly is critical here. That's an obvious case. Another case is as an example of using particles as an assay readout. So one example would be as a biomarker experiment. So we're detecting particles and their concentration is the test result itself, right? So the better you can measure the concentration, the higher your sensitivity and specificity will be until that's no longer a limiting factor, right? So these are two very common sort of broad examples of where controlling the particle concentration and measuring it better is critical for tightening up experimental variability. So Spectradyne's technology allows for that kind of control on concentration. And so let me tell you a little bit about that for a second. This is our product here. It's called the NCS1. It's a benchtop instrument. It's about a foot wide, so it's pretty small on the benchtop. This is new technology, and it delivers accurate concentration and particle size starting at about 50 nanometers diameter on the small end and going up as big as 10 microns in diameter on the upper end. Importantly, this technology uses electrical detection means, so it's not an optical technique. That's very important for delivering the accuracy that it does. It's microfluidics enabled. What we're showing here is a microfluidic analysis cartridge that takes just 3 microliters of your sample. So overall, the system's easy to adopt, it's easy to use, fast, results in minutes, and affordable with also saving you sample costs. It's used in many different applications. We've got customers using it for quantifying extracellular vesicles, EVs, also measuring virus. This is a timely example now, and bacteriophage, A lot of our industrial customers use it to detect protein aggregation by quantifying the aggregates when they're smaller, and so sooner in the aggregation process. Gene therapy applications, et cetera, et cetera. So long list. We've also measured examples of industrial materials, paints and floor finish and things like that. So because of the electrical nature of the technique, the measurements are agnostic to the particle material. So it's a broad range of applications here. So how does that work? So the underlying technology is called resistive pulse sensing. That is essentially the Coulter principle. And it's been the gold standard for decades for measuring larger particles and cells in the Coulter counter. What we've done is brought that into the 21st century by scaling it down and putting it in a microfluidic cartridge. And that allows a bunch of other usability features that allow us to measure smaller particles than the traditional Coulter counter can. And in this technique, particles are measured one by one, very, very quickly, thousands per second. And we measure the concentration and the size very directly. So in contrast to optical techniques, where the size is inferred from its Brownian motion, for example, or where the concentration is inferred by geometric assumptions, we're not doing that here. So let me explain how that works. What we do is we flow the whole sample through a bottleneck in the fluid path and run an electrical current through there as well. And when a particle goes through, it blocks some of that electrical current and gives us an electrical signal each time. So this is the signal and actual data recording of a single particle passing through. This is very short time scale, obviously. And so every time one particle goes through the constriction, we measure a number of things, but two main parameters. One is the amplitude of the signal we get. That tells us how big the particle is and how fast it went through. And that tells us the amount of liquid that's flowing through the constriction so that we can know the volume of sample that's been measured. So every time a particle goes through, we measure how big it is and how much of the sample has been measured. And that's counting in a volume that's directly concentration measurement. So that was the signal from one particle detection event. This is a longer acquisition, a 1 minute time trace. And here all the particle events we're highlighting by red dots. And you can see that there are 4 different sizes of particles in the sample. Small ones down here that only make a small spike, and big ones up here and some bands in between. We can just directly from here read off a histogram of counts as a function of amplitude, right? And remember, because we know the amount of volume that we've measured of sample, we can turn these counts into actual absolute concentration numbers. So this is what our final data output looks like. This is a plot showing the concentration on the y-axis versus particle diameter on the x-axis. And this particular sample was a mixture of polystyrene standards, 50, 90, 120, and 150 nanometers in diameter. And you can see that they're all clearly resolved and quantitative, right? You can measure the concentration over any size range here. This kind of resolution and quantitative concentration is not obtainable by any other method currently. I want to show you just a quick video here that gives an overview of using the instrument. And it's kind of fun to watch too. Here's the user loading up 3 microliters into the cartridge. Cartridge goes in the instrument, and it's all a very automated process. After this point, the instrument is all software controlled. So here what we've done, I'll pause it for a second. What we've done here is taken one of the microfluidic cartridges and put fluorescent particles in it, and put it on a microscope, inverted fluorescence microscope, to see what's going on inside. And so this is just showing fluorescent nanoparticles doing Brownian motion inside the cartridge. And now we're looking at some of the features inside the cartridge. So this serpentine pattern here is one of the engineered parts of the cartridge. It's an embedded filter. We'll see it in action in a second. And then we're going to release the particles into the cartridge. So they're going to flow from right to left here. And as they flow, you can see this filter, which is on the right-hand side, collecting large particles that would otherwise block the small sensing constriction. So it's working immediately. And that allows very smooth operation in very complex samples like plasma, for example. Now here are the particles going through the constriction, which you can't see because it's a dark field image, but it's right here. And every time a particle goes through here, we get an electrical blip that tells us how big the particle is and how fast the flow rate is. And as you can see, we do this very, very quickly. So we can get an accurate particle count and concentration size distribution very quickly. So I want to just make one comparison to a common method out there. It's called nanoparticle tracking analysis. You've probably heard of this already. And a lot of this information is available on our website too, so I encourage you to go and check it out. Here we have a very full library of information, publications and stuff. So this example is about extracellular vesicles. This was an EV prep that I believe were urinary EVs. And we measured this same sample using 3 techniques. One was our NCS1 in blue. 2 is cryo-EM, which is the gold standard for doing this, measuring this kind of sample. And then, but it's very time consuming, obviously. And then NTA, which is commonly used. And what we see here, and we can talk more about this later if you like, if you have questions, but what we see is essentially, excellent agreement between our technique and TEM, the gold standard, all the way down to 50 nanometers diameter. But what we see, this measurement exposes that the sensitivity of NTA, nanoparticle tracking analysis, is rolling off to smaller sizes. And this is really just a fundamental physics limitation of the technique because the small particles don't scatter light nearly as strongly as the big ones. And there are many ways that you can show this yourself, even in your own lab. And we have information about that on the web, on our website, if you like. But so what we're seeing here is that NTA is falling short by 4 orders of magnitude at least. This is a log scale on the y-axis, right? So 10,000-fold error concentration or concentration error right in the sweet spot for measuring EVs. This is exactly where people want to be measuring their EVs. And so this is a critical failure mode essentially. But the NCS1 is measuring accurately, matching with TEM the whole way down, right? This has been shown with other techniques as well. So at this point, I would like to turn it over to Zach Troyer from Case Western. He is a 5th-year PhD candidate in the Tilton lab there. And he's going to talk about how accurate concentration measurements helped his research, and specifically about how to purify their EVs better. So bear with me while I do this transfer here. One sec.
So hi guys, I'm Zach Troyer, as was stated. I'd like to thank Jean-Luc and Lou for giving me this chance to give you guys a couple slides about how knowing the concentration of my extracellular vesicles has helped me. So my research is, as mentioned, extracellular vesicle based, so EVs. And I look at how extracellular vesicles can have biological effects on cells and cause cellular responses in general. So I'm just going to give you a couple of slides about how knowing the concentration of my particles helped us to establish a better isolation method of our extracellular vesicle preparations. So what you can see here is 6 independent biological preparations of extracellular vesicles. So these EVs were made the same way from the same number of cells 6 different times. And the important thing to take away here is that while on this size distribution plot, which does show concentration and size distribution of our vesicles, while we do see agreement amongst the biological replicates in terms of the general size distribution pattern, we do see differences in the concentration, which is kind of represented by y-axis shifts between the different independent biological replicates. So as scientists, we tend to assume that a biological replicate is an accurate way to have multiple types of samples of the same thing. But what you're seeing here is that amongst biological replicates, there are differences specifically in the concentration. And I think this is perhaps better represented by this bar graph that just reports the total EV concentration between 65 and 400 nanometers of size. And here again, you can really see that there are differences amongst our biological replicates in terms of the concentration. So you know, without having some sort of instrument to measure concentration of our extracellular vesicles, we wouldn't really have been able to catch this and normalize for this in our downstream assays. So given that we do have an instrument to figure out the concentration, we are able to then go forward with our biological activity assays in cells and normalize our input of extracellular vesicles between our different preparations. But that's not really the end of the story. So here, this is an EV functionality assay where we're looking at a cellular response in response to extracellular vesicles. And we've taken 3 of our biological replicates of EVs and added 2 different doses of those EVs to our cells to generate a response. And we've obviously at this point normalized our EV input using a instrument that measures concentration. And what you can see here is that while 2 of our independent biological replicates do agree in terms of the functional response they're getting from the cells, our 3rd biological replicate, even when we've normalized by how many EVs we've added, we're still getting a differential response. There seems to be some sort of variance going on. As you can see, they're highlighted in the circled by the red. So before, if we had not had an instrument to tell us the concentration of our extracellular vesicles, our first thought might have been, perhaps I added too few EVs of this EV6 condition, and that's why I'm getting a lower response compared to the other 2 biological replicates. But after having an instrument that can measure concentration of extracellular vesicles, we can be sure that we added the correct number of vesicles to this condition. And thus that tells us something about this independent biological replicate. It tells us that these EVs are, for whatever reason, different than the other 2 biological preparations, even though, again, we added the same number of particles. So there's some sort of variance going on here. And having the concentration of our particles allowed us to detect that variance and know that something is going wrong. And in my case, we suspected that this variance was coming from difference in the way that we harvest our extracellular vesicles. So we wanted to basically switch to a new method and see if we could get better responses and get extracellular vesicles that were working better in our assay. And we were only able to decide to switch to this new method because we recognized that there was an issue through knowing the concentration. So here you can see the data, the same sort of assay as before, but we've now switched, we've added in a new method of harvesting EVs that's gentler on the extracellular vesicles themselves. And the important thing to take away here is that when you look at this point here, we now have extracellular vesicles that are generating a much better response in our assay. And we can even add less of them, and we're still getting a better response. So, you know, we only added 10 to the 9th particles here, and we've added, you know, 3 times 10 to the 9th particles here, but our new method produces EVs that are much better at generating response in our assay. So we have corrected our issue, presumably, of the harvest method of our extracellular vesicles causing issues. And we were only able to do so and figure out there was an issue by, again, getting accurate EV concentration reads using an instrument. In this case, we used the NCS1. So yeah, this is just kind of a good case study of how knowing the concentration was really important to improve our research and make our data look a lot better and tighter. So with that, I'll throw it back to Jean-Luc.
Great, thanks a lot, Zach. So we're hoping to see that one in print pretty soon. And in the meantime, let me share with you a couple of other slides also, or a couple of other publications that have been out recently. Let me make sure you're seeing, yeah, good. Okay. So a couple of recent ones. One is from, this one is from UCLA, a Kalanick group. These folks used the NCS1 to quantify their EVs also, and what they found was interesting because in prior publications they had used NTA, and based on that data they thought their EVs were all over 150 nanometers big. But as I showed you before, that actually was an artifact of their measurements. And so here they were able to realize that, in fact, the majority of their EVs were below that diameter. So this is an interesting example of better measurements making for better science, basically. In nanomedicine applications, this is a pilot study we did with Pfizer in New York. And in this case, we measured nanoparticles directly in mouse plasma. And you can view this poster in our library also. This is an example of measuring nanoparticles. I think these were 100 nanometer particles that were used directly in very complex media. And that's only possible because we're using an electrical measurement technique that measures particles one by one. So we can measure in very complex polydispersed samples. Another example from Bristol Myers Squibb, using the NCS1 to quantify protein aggregation. This is another interesting comparison, technology comparison between our NCS1 and NTA and other optical techniques. And finally, one other example from CRO in Germany called Coriolis Pharma. These researchers measured a variety of samples across different techniques and found that the NCS1 was very diverse, in its ability to measure different samples well. So in summary, Spectradyne's NCS1 provides accurate concentration that helps you do better science, basically. It helps improve product safety and efficacy. And as Zach showed us, it can reduce the process variability and the measurement variability in your experiments that helps you do R&D faster. It's fast and easy to use, and please contact us if you'd like to learn more. One of the things we do to demonstrate our technology is to measure samples for free for folks as a demonstration. So give us a call, send us a sample. We'd be happy to show that for you too. So we're going to open up to questions at this point. And as a reminder, there are two ways to ask. You can use the Zoom chat tool, or you can email Lou directly. And I will transfer it over to Lou, or I will unmute Lou here 1 second. Okay, Lou.
Okay, all right, great, thank you.
So the first question is from Madeline Strain. What is the optimal concentration range that the NCS1 can detect? So the overall sizing range of the technology spans from 1e⁴ or so particles per mil up to 1e⁻11 particles per mil or so. So it's a huge dynamic range of concentration. It depends specifically for your sample on what size range you're interested in measuring, but generally that's the range, and it's much broader than other technologies also. The second part of your question was about whether the instrument requires a blank to compare the samples to. And so measuring blanks is a really important question, mostly from the perspective of, if you're diluting your sample into a diluent that contains particles itself, then you're going to essentially, at some point, you'll dilute your sample below the background of particles in your diluent. And so it's important to know what your background concentration of particles is, but there's no blank normalization required nominally for any measurement. So you just put your sample in, and it counts the concentration of particles in that sample, and then you grab another sample. So there's you don't have to calibrate to a blank every time if that's what you're after. Other questions?
I've got one here for Zach, I believe. You talked about using different EV harvest methods, and that different methods you showed produce different levels of functionality. Can you comment on which methods you've tried and your experiences with them?
Right. Yeah. So some of the methods I've tried is one is ultracentrifugation into a sucrose cushion. And then we followed that up with size exclusion columns to purify And that's one that we've seen generally the EV yields are not too good. And I think it's probably the ultracentrifugation damages a lot of the extracellular vesicles. And then putting them through size exclusion columns will also dilute them to an extent. So it's kind of not the best method. And just ultracentrifugation in general is probably not very good on the EVs. Ones that have worked really well for us, one specifically is tangential flow filtration methods, or TFF, in which the EVs kind of, it's very gentle. They just pass through a cycled tubing system where they go through a filter, and then there's pores in that filter, and small protein aggregates and things smaller than EVs can pass out, and it'll basically concentrate your EVs over time, but it's very gentle on EVs, and that's one that we found works very well.
Okay, thanks. Looks like Rob Damitz, hopefully I pronounced that right, has his hand up, so I'll unmute you. Can you talk?
Yeah.
Okay, cool.
Yeah, the chat doesn't appear to be working. I can't send my question, but so I'm more familiar with background membrane imaging. That's what we currently use. Can you maybe, if you're familiar with it, can you speak to the advantages and disadvantages of the two techniques?
Background membrane imaging, is that like filtration and then imaging on the membrane? Is that how it works?
Yes.
Okay, and so I guess what size range of particles are you accessing with that?
Yeah, is that specific to USP 788 or?
Yes.
Okay, so that's a completely, so that's 2 microns and larger, and we can go up to 10 microns. We detect when we do comparisons between, say, flow imaging and or membrane microscopy with protein aggregates, we always find more, and that's because that we're not reliant upon background optical contrast, which those other two techniques are. And as you know, the protein aggregates don't tend to have a lot of contrast. They tend to be fairly transparent, so there's great difficulty in seeing them. Mm-hmm. Beyond that, I think the other point that we like to make a lot, and we've got some interesting data we can share with you, that if you look for protein aggregates at smaller sizes, you'll detect it much earlier. And I mean, basically, by the time you're seeing aggregation at the 2 micron and larger size, you've got a pretty bad formulation at that point, or you've stressed it to a very high degree. And we can actually see aggregation much earlier, down in the 100 nanometer range, and save you time as far as if you're doing pre-formulation, by showing you that that aggregation much earlier than you would ever see it with the types of techniques you're talking about.
Okay, great, thanks.
I think I have another question here. Someone noticed that on the list of applications for the NCS1, there was something about viruses. Can you comment more on typical applications related to viruses, which I guess is a subject near and dear to everybody right now.
Everybody, yeah, sure. So one of the advantages here is that we can count virus particles directly because they're biological nanoparticles, and we can count them in complex media like cell culture media or plasma. And we can do that very quickly. So rather than doing live biological titer, for example, which can take hours at best, or even days in some systems, we can just measure, count the particles directly in minutes. So saves a lot of time that way. So our customers in gene therapy applications are obviously excited about that. And they can also use it to assess the purity of their samples at various stages of production. So one case I'm thinking of, I don't have the slides here, but they're available in our library on our website. shows that we can see a peak, viral peak in the size distribution emerge as the purification progresses through different steps, different stages. And so this tool becomes, the NCS1 becomes a tool for quantifying the purity of their viral preps that way as well. So hope that answers your question. Maybe we take one more because we are past the 30-minute mark at this point, and then any other questions,
here,
you can certainly email us.
Oh, hi. So I missed the first couple of slides. I'm sorry if this is a repetitive question, but are there any limitations in terms of charged particles, just given how you're using electrostatic changes to measure the different sizes?
Yeah, good question. So we have not seen any effect due to charge on the particles. In principle, it should not affect our measurement. One of the things that's happening in the sample is that charged particles are screened by ions in the conducting medium, so with the Debye layer, and so overall the particles closer to neutral. So we haven't seen any effect from charged particles. Does that answer your question?
Yeah, sounds good. Thank you.
Great. Yeah.
So you want to wrap up, Jean-Luc? Just
Sure. Yep. So thank you all for your time. Thanks for joining us. Please feel free to reach out to us with more questions if you have any. We'd love to hear from you, and come visit our website. And I hope everybody stays safe out there. Yep.
Thank you all. And as Jean-Luc said, stay safe.
Great. Thank you very much. Thank you, Zach.